this is already on.
Yep.
Maybe that was too fast.
Can you do the whistle thing?
I can't. Could I? Right. So right now I'm working on whole genome bisulfite sequencing on C. elegans and Oscheius Tipular. C. elegans, just a little background, are non-parasitic roundworm. They're about 1 millimeter long. They're widely used for neuroscience studies because they have a very similar nervous system compared to ours, and they're often used in like behavioral studies and such, as well as we do culture cells with them. And then Oscheius Tipular is Riley's project. He's a graduate student that I'm working with. He collected transcriptome data, and now with this bisulfite sequencing, we're hoping to pull some useful information out from that. So before I go into bisulfite sequencing, just a little bit on epigenetics. Epigenetics is very important. It helps us understand twin studies when one twin will get cancer and the other one doesn't. So it helps us understand what's going on at the molecular level. So epigenetics is the phenotypic change that happens on a DNA. So instead of having a genomic DNA sequence change that causes behavioral shifts, instead it's just how the DNA is regulated, and it doesn't change its structure at all. Epigenetics often happens as a stress response to environmental situations. And right now, publications-wise for C. elegans and KJO data, we could not find any methyltransferase genes in C. elegans like we do in KJO when we ran our transcriptome data. So all that we know publications-wise is that there are histone modifications, but we don't know if there's necessarily methyltransferase genes, which is used for determining whether or not methylation is occurring to help influence epigenetics. So DNA methylation is just the addition of a methyl group, and it's influenced by the methyl or DNA methyltransferase genes. There are other modifications that can occur in epigenetics. Histone modifications is one of them, and I listed a variety of those, and then non-coding RNA, which I'm not very familiar with, but yeah, still can have an influence on epigenetics. So what I'm trying to do with my C. elegans and Oscheius Tipular is I'm trying to find these methylation sites to see if they actually can methylate their DNA or not, because we don't know that yet. So first, our methods are pretty standard compared to other papers that have been looking at this stuff. We just collect our DNA, we fragment it with fragmentase enzymes, which we just got some new stuff of. So we're kind of stuck on, we're on this part right now. But other papers have been using sonication methods, which is just a more mechanical way of breaking down the DNA versus enzymes. Then what we do is we do our 3' pair adenylation. So we kind of patch up our ends after the methyltransferase genes have gone through. We aligate adapters because what we plan on doing is sending it to Illumina for HiPut sequencing to find these methyltransferase genes. So after I put the adapters on the DNA, what we do is we run all that genomic DNA out on a gel then, and I select for the 200 base pair size DNA because it works well with Illumina when they go to sequence their DNA. And the final step is actually trying to find those methylation sites, and we use the EasyDNA methylation gold kit from Zimo. Yeah, it's a common, it's a pretty common kit for finding methylation sites. They pretty much, yeah. So this is just kind of an overview of bisulfite sequencing. You'll notice that we fragment the DNA, we repair the 3' ends, and then we take them with adapters. And at the bottom, you'll notice that there'll have to be 2 adapters ligated on both the 5' and 3' ends. And this is just for Illumina sequencing when they do their bridge amplification steps. So our main goals for this project is to be able to send these samples off to Illumina so we can get our sequencing. And then Riley had collected some transcriptome data, so we're hoping that we can compare our methylation sites to his transcriptome data to see where these methylation sites are. And then we're pretty much just going to analyze the different variations of methylation that happens. This will be technically our wild type sample. We'll have a few more samples going off after doing an actual stress response, such as heat shock, to these worms. And then, yeah, so right now we're troubleshooting our adapter ligation step. For some reason, we can't get our adapters to ligate onto our DNA. We're not quite sure what's going on with that, but if we can't get this step figured out, we might just send it off to have someone else sequence it and then send it off for Illumina sequencing. But any questions?
I have a couple.
Sure.
First, the purpose behind it, what's the ultimate goal, what would you do with it? And second, if it's phenotypes, that means this is something that would be inherited to the next generation.
Okay, yeah. Epigenetics is heritable to next generation uses. A previous project I have worked on under Dr. Lucia Carvelli was looking at whether or not amphetamine addiction was heritable, and because there wasn't a genotype change on the gene level, but we still were able to see behavioral changes through several generations, we had to conclude that it was possibly an epigenetic change that was modifying the gene expression to cause those behavioral changes down the line. And what was your first question?
What's the purpose of it? What's the ultimate goal with that? If you get results, what would they be used for?
Sure. C. elegans, they have multiple uses. So in the ecology lab I work in, we analyze soil nematodes and how they interact with their environment. So there are many different types of nematodes, and C. elegans is just one of them. But C. elegans, they go through like freezing periods, heat shock. There's dry periods where they have to regulate their genome to survive in that type of environment, and that's really important to understand the mechanisms behind that. Otherwise, yeah, there are actually health science related stuff going on because they are one of the smallest, simplest organisms with the nervous system. It's really important that we look at these methylation sites so that when we're doing behavioral studies on them, we can better understand what's going on at the molecular level.
Dumb question. What's the
Methylation
methylation site?
site?
Yeah.
It's just when we attach a methyl group onto DNA, and that'll regulate whether or not the gene is expressed. And if a methyl transferase enzyme brings in a methyl group, what happens is we'll see a decrease in expression. But we can also have the opposite effect where it'll demethylate and you'll see more expression. So.
Can I ask just a general question?
Sure.
So since you were in development, I get to ask you to put this together. So when you're thinking about your environmental response to stress model with the C. elegans, where do you see broader implications for other organisms besides just the nematode?
The nematode, yeah. Like I said earlier, they are widely studied for neurology reasons. So we can correlate biomarkers between their nervous system and ours. So if we find nervous system methyltransferase genes, we can correlate like behavioral studies such as addiction, and we can follow that through many generations to see if we can find more biomarkers for health science reasons in trying to help like cure addiction and understanding diseases like that.
So apart from the whole addiction thing, just looking at environmental stress responses like the ones you talked about, like desiccation or drying or something like that.
Sure.
What are the broader implications for types of situations that organisms might face in the environment?
Oh.
Not just the people part.
not just the people part. Organisms in general, they have to adapt to their environment, and we find that with a variety of organisms. Before we'll see natural selection happen where genes are selected for, we'll see epigenetic responses for short periods of time, just to see if they can kind of toughen out through the time, because it's kind of taxing on an organism to have to go through natural selection and repopulate the area, I guess. Yeah. Did that answer your question? Is there
It's
any...
getting closer.
Okay. So keep asking questions.
That's okay.
Okay. Yeah.
I had a question,
Sure.
and I apologize if this is off the mark a bit, but
That was fun.
so with these worms and how they respond to environmental stress, does that have any impact on the soil, and if so, what?
That's a really great question. I'm assuming it does have an impact on the soil. Although they are non-parasitic, free-living roundworm, they do feed off of certain microbes in the soil. So that would probably have a pretty big effect on agriculture in general, or just the overall health of the soil. Yeah. Other than that, I'm not quite sure, but I should definitely look into that one. Hi.
So, a warming of the atmosphere, eventually warm soils, which would be a stressor on these kinds of organisms. How would that ripple through the
So what we find with nematodes is that they're pretty tough. And they'll actually slow down their reproduction and their growth dependent on the temperature that they're at. So if we see a warming of the soil, we're expected to see a faster growth rate and a faster reproduction time with many of these nematodes. So in areas of desiccation, they do have to have some sort of fluid after a while. I mean, they'll definitely try to tough it out, even possibly go into dour stage, which is their survival mode. But I would assume that if they can't use their epigenetic responses to help them get through that period of time, I'm assuming that they wouldn't be able to stay in that area.
I have a question. Does the density of population of these worms, or is it a positive or negative correlation with the richness of the soil?
Nematodes, there's a variety of them. And we have nematodes up here that they don't necessarily have in Kansas, or they'll have like a different strain of them. So if we're just looking at a certain area, I'm assuming that the more rich the soil is, we'll find more of these nematodes, just because the environment's better able to equip them with the stuff they need to reproduce and survive.
But do they enrich the soil themselves?
They enrich, I don't know if they enrich the soil themselves, but I'm certain that they have an ecological effect in, the grand scheme of things with compared to other organisms and bacteria that live in the soil. Yeah.
You kind of answered my question, but on the different strains, like I ran gels and diabetes research on the different strains. We used mice.
Okay.
I was just curious about the different strains. Do you work primarily with one strain, or do you compare different strains?
Yeah, so in ecology we'll be comparing different strains, and we'll be kind of dissecting a little bit more about what these nematodes are and what they do for soil. However, C. elegans is our model organism. We don't find other nematodes being used currently, but possibly that could change, especially now that we're doing a lot of more transcriptome data. And these nematodes are very similar, but they can vary just a little bit. And like I said, we were able to find methyltransferase genes in KGO, but we couldn't necessarily find that in C. elegans. So that might be a limitation of that model organism, and we might need to switch over to a new model organism that still has the simple structure and same biomarkers, just a little bit more of what even health sciences need, I guess. So. Anything else? Okay. Well, Well, thank you so much. It's been very